Hello everyone, welcome to yet another Working Group 2 webinar where we again have two very nice speakers that will present a little bit on the research that they do in their groups and also in the context of what is relevant in Working Group 2 of this cost action, of course. So our first speaker today is Dr. Javier Cabrera, who is a plant biologist at the Centro for Plant Biotechnology and Genómica in Madrid, where his research explores how soil microorganisms reshape the roots development. Throughout his career, most of his contributions have focused on the molecular mechanisms underlying plant-nematode interactions, particularly those involving root knot nematodes and the development of nematode feeding sites in the group of Carolina Escobar. He later moved into the study of lateral root development in the group of Miguel Ángel Moreno-Risueno, and more recently also worked with Juan Carlos del Pozo on beneficial fungi as drivers of root system improvements. He now leads his own research line on plant-microbe interactions, seeking to connect fundamental discoveries in root biology with sustainable and translational applications in agriculture. So without further ado, Dr. Cabrera, the floor is yours.
Thank you very much. Thank you, Naomi, for the introduction. And thank you very much to the coordinators of the Root Benefit Cost Action for organizing these seminars and giving us the opportunity to present our group and our work that we are doing here at the CBGP, the Center for Plant Biotechnology and Genomics in Madrid, this building, investigating the development of the root system, specifically during the interaction of the plants with the microorganisms in the soil. This is a new research line within the group of root response, in which specifically we want to understand how the endophytic fungi are able to alter the molecular pathways leading to the formation of the lateral roots. This is a new line, as I said, within a main research line led by Dr. Juan Carlos del Pozo and Dr. Elena Caro, focus in the transcriptional and genetic regulation of the root development in the context of abiotic stresses like the heat stress or the phosphate starvation or the combination of both, and how these stresses also affect the microbiome in the soil and how these changes in the microbiome also affect the root development. This is all the people from the root response laboratory. Our starting idea is that plants with a more developed root system will be able to cope better with these multiple and unpredictable stresses that are derived from the climate change. As you all know, the root system plays essential roles for the plant survival and development. They anchor the plants to the soil, providing them with mechanical support. It is the main compartment for the storage of the photoassimilates from the photosynthesis, and it is also in charge of the water and nutrient uptake to the aerial parts of the plants. All these functions, very important for the plants, are today compromised because of the presence of these multiple and unpredictable stresses derived from the climate change, and also because of the reduction in the use of chemical fertilizer to achieve more sustainable agriculture. This, in the context of the growing population that we will have at the end of the century, is some problem that must be addressed. Thanks to the molecular research made in Arabidopsis thaliana, we have a huge knowledge of the molecular and hormonal mechanisms regulating the root development. Briefly, the roots can enlarge by the growing of the main root through the root apical meristem or through the appearance of lateral roots. In the root apical meristems, the cells start to divide and to differentiate in the different cell layers. And we have a lot of molecular information, as I said, with multiple transcription factors and almost all the phytohormones regulating this process of divisions. And the same can be said for the lateral root formation. There has been defined different checkpoints during the lateral root formation. The first of it is this oscillation zone that can be visualized with the DR5 luciferase transgenic line. This is an area very close to the root apex where we can find up and downs in the auxin signaling, and each peak in the auxin signaling can mark a pre-branch site in the main root, and some of these pre-branch sites will later divide in the silent pole pericycle to form the founder cells. This founder cell will continue dividing to form the lateral root primordia. As I said, there is a huge molecular knowledge of this process of the lateral root formation, and the main one, and that we are going to talk later, is the auxin signaling pathway leading to the formation of the lateral root. As I said, all this vast knowledge of the molecular mechanisms regulating the root growth has been achieved in Arabidopsis, but the problem is that most of the time it has been achieved in Arabidopsis isolated from their environment, growing in vitro and monoxenic culture conditions. And this is what we are trying to fill this gap with our research, because as you all know, in the field, in the root system cohabit in the soil with a plethora of microorganisms, including fungi, bacteria, viruses, nematodes. And we think that answering this question of how soil endophytic fungi modulate the plant developmental programs to reshape the shoot architecture, and specifically how they do it at this at these control points of the lateral root formation, specifically the pre-branch sites, the solutions, and the founder cells, will give us not only fundamental knowledge in the fields of root development with the discovery of new molecular pathways that only arise when there is an interaction with microorganisms, or in the fields of plant microorganisms interaction, but also a huge applied knowledge that can be easily transferable to the industrial partners, supposing an alternative to the use of transgenic and a reduction in the use of chemical fertilizers. So with this framework in mind, we settle different questions, research questions, to go from the discovery of beneficial plant-fungal interactions to the translations to crops and the industrial partners as biostimulants. Of course, in the middle, we have all the basic knowledge that we can understand in the fields of root development and plant biotic interactions. So the first question was the discovery of new plant-fungal interactions that benefit the expansion of the root system and the increase in the number of the lateral roots specifically. We started our study with a collection of endophytic fungi isolated from wild population of Arabidopsis thaliana belonging to Dr. Soledad Sacristan here at the CBGP that was published already in 2013. Now we have established another collection of endophytic and rhizosphere bacteria and fungi isolated from plants that has been exposed to combined or individual heat and phosphate deficiency stress. But I'm going to talk later about this collection. We started our research a couple of 3 years ago with this collection from Soledad Sacristan. We have performed a screening in Arabidopsis plants grown in vitro. And after incubation of the plants with the different strains, we measured the main root length and the number of lateral roots. We obtained interactions very detrimental for the plant growth, other ones that were neutral, and some of them with a positive effect. In particular, we selected 2 fungal strains, either 2 and H2, because they enhance very visually the plant root system development. I want to center mainly in the results that we obtained for H2. H2, we knew later in sequencing the molecular markers that it belongs to a strain of Penicillium mellini. As you can see, when we incubated Penicillium mellini with Arabidopsis in vitro, we obtain a larger main root and a higher number of lateral roots, and the lateral roots seems to be larger also. So at the end, we obtain a more expanded root system than in the mock plants. We had the opportunity to transform this Penicillium mellini with GFP and observe that it colonizes the whole root system along the whole root, but only at the superficial layers of the root, at the epidermis and cortex, what for us seems to correlate with an endophytic lifestyle of this fungi, because it does not penetrate into the vascular cylinder. So it's endophytic, it seems to be beneficial. We could expand this beneficial effect of our strain of Penicillium mellini to the whole species, Penicillium mellini, because we ordered different strains for around the world from the CBS and obtained similar beneficial effects when we incubated with Arabidopsis or with tomato. In conclusion, we have now a very good candidate to study the root development with our plants. So the second question and the fundamental knowledge that we want to generate is to understand at what developmental stage this fungus is altering the root development, and how at the hormonal and molecular level and atomic level is affecting the root development. To answer the first question, we incubated the line, the DR5 luciferase, with which we can observe the oscillation zone and the prevalent sites with Penicillium mellini. And what we observed is that there is not a high induction of the auxin signaling through all the root system, but specifically we obtain a higher number of prevalent sites in the main root. And specifically, we observed an induction of the auxin signaling in the oscillation zone. To understand at the molecular level what is happening, we obtain first the transcriptome of the whole root system of this interaction at 3 and 6 days. We were very surprised because we obtained a very mild response, transcriptomic response for the plant, probably because, as I said before, the fungus is not penetrating in the vascular cylinder, so it seems to be recognized as a beneficial microorganisms. First, we observed that almost there are no regression, only upregulation, and then at 6 days, there is a balance between upregulated and downregulated genes. Also, surprisingly, there are no almost common genes between the two stages, meaning that the response is very specific at first, and then it changes with the interaction. When we obtain the gene ontology term enrichment analysis of these differentially expressed genes, we observe categories like this of the lateral root formation enriched among those genes upregulated at 3 days, with genes like the LBDs. This is very important for us because it means that very early during the interaction, the fungus is altering the lateral root development. We also observe the response to other organisms induced at 3 days, but this response is rapidly enriched among the downregulated genes at 6 days, also correlating with a beneficial endophytic fungus lifestyle. The auxin homeostasis that seems to refer to a specific auxin signaling in specific parts of the root is maintained as upregulated during the whole stage that we studied. As I said, the canonical auxin signaling pathway leading to the formation of the lateral root seems to be altered during the during the interaction with the fungus. So we perform interaction tests with mutants for this auxin signaling pathway. We observe that we obtain our induction in the number of lateral roots with called 0. However, in the solitary root mutant that is very up in the pathway, is a repressor of the cascade, we could not observe any induction in the presence of Penicillium melini. However, it was very surprising that in the double mutant ARF7, ARF19, that are key regulators of the response of the or LBD genes, and that do not present any lateral root in the mock treatment, or almost no lateral roots, there is a high induction in the presence of Penicillium melini. And the same can be said for the double mutant LBD16, LBD18, that yield an increment in the number of lateral roots similar to that obtained in the cold zero plants. So these results seem to say that the Penicillium melini is activating alternative or redundant pathways, converging in downstream effectors, but bypassing these transcription factors. To understand better what is happening specifically in the oscillation zone and the pre-branch sites that, as I said before, seems to be altered during the interaction with Penicillium melini, we have obtained the transcriptomes of these pre-branch sites, oscillation zone, and root apex. And at the first, we were very afraid with this experiment because we didn't know if we will be able to separate the transcriptome of these three parts that in Arabidopsis are millimeters. But our analysis seems to confirm that we have obtained transcriptomes different between the root apex, the oscillation zone, and the pre-branch sites, as you can see in this principal component analysis. This is an ongoing work. We are now with the help of Laura Gutierrez and Francesco Blasio, analyzing the genes differentially expressed in this oscillation zone to know if we can correlate the results that we obtain with the DR5 luciferase with the transcriptomic changes and see the molecular mechanisms underlying this induction of the auxin signaling. At the same time, we performed the hormonomic analysis of the whole root system of plants incubated with Penicillium melini at 3, 6, and 12 days with the collaboration of this group in the Czech Republic. As we observed in the transcriptomic response before, there is a mild induction with no induction of any of the main phytohormones like IAAs, cytokinins, salicylic or jasmonic acid. But what we observe is a young accumulation at 6 days, mainly of indole compounds related to the glucosinolates biosynthesis, what correlates with the defense downregulation that we observe in the transcriptomic analysis. We want also to know what is happening from the fungal point of view, let's say. We have obtained the genome by Illumina and by Oxford Nanopore to know what genes or metabolites can be synthesized by the fungus to create this beneficial effect. We already have a draft of the genome with the Illumina sequencing in collaboration with Dr. Soledad Sacristan and Dr. Sara González Bodí and Gonzalo Monteoliva. But now we have the sequencing with Oxford Nanopore, and we are trying to complete and to polish this genome to basically center in these effector proteins or metabolites from the secondary metabolism to understand that if the fungus is secreting something to the plant that can be altering the root development. And also in the field of study of Juan Carlos del Pozo and Elena Caro, we have obtained the single nuclei RNA-seq of the roots and the small RNAs and the methylome by whole genome bisulfite sequencing of the interaction. So we think that at the end, we are going to have a vast knowledge of the molecular mechanisms that has been altered during the interaction with penicillium. But as I said before, it's very important for us that this increment in the root system of the plant can be conferring a beneficial effect to the plant to cope with different stresses related with the climate change. This again is part of the work of the group by Juan Carlos del Pozo and Elena Caro. As I said before, they are specialized in heat stress, in phosphate starvation, and how they alter the root development and the microbiome in the soil. The phosphate starvation is relatively easy to apply to the plant by just taking out the phosphate from the media or from the fertilization that we use for the pots. And we have already in the thesis project of Ivan de Besa, very good results for the fungus that I said before that we obtained from the collection of Soledad Sacristan, IDA2, where we can observe an increment in the root system and also in pots in the aerial part of the plant. This is an ongoing work, and just because we don't have enough time, I prefer to center in the project of Penicillium melini. But the heat stress, we think that can be refined to better mimic the field conditions. In laboratory, usually what we do is to put the chamber at from 22 degrees to 32 degrees to apply the heat stress to the plants. This in the in vitro chambers or also in the greenhouse. This is heating at the same time the aerial part of the plants and the root systems. However, in the field, in natural conditions, the soil could produce a geothermal buffer effect to the root system with a temperature gradient in the roots from the cooler from the deeper layers of the soil to more warm and more similar to the ambient temperature in the superficial layers. So with this in mind, Juan Carlos del Pozo a few years ago engineered this system, the TG roof, to simulate the temperature gradient that we observed in the field. In this system, that is basically a metallic box, we can put the plate, the Arabidopsis plates here. The roots are growing in dark, as we always do, in darkness, and the aerial part is in the light at the same temperature of the chamber. However, there is a water cooling in the bottom of this box, so there is a gradient of temperature similar to that I give in the soil. This is more graphically visualized with a thermal camera. This is a plate growing at 22 degrees with the upper part of the plants and the roots at 22 degrees. This is at 32 degrees, and this is the gradient created by the DG roof. And very importantly, this is translated to the root development. This is how it looks a root of Arabidopsis as we all grow in the lab. This is how it looks with 32 degrees at the same time in the area and in the roots, with a very huge affected phenotype for the roots. And this is in a more natural conditions using the DG roof that is more similar to that of the 22 degrees and not so dramatic like this of 32. And the same happens in the area part of the plant. Juan Carlos already engineered this for pots and for rhizotrans to use them with crop plants in the greenhouse. And with this, they have created a collection of endophytic and rhizosphere bacteria and fungi isolated from plants exposed to this heat stress by using this gradient and to phosphate stress deficiency in combination of both stresses or individually. They are now studying this collection of fungus and bacteria, and there is a lot of predoc students and postdoc involved in this project. And to finish my talk, as I said, our last goal is to translate our basic knowledge and molecular knowledge to crop plants and to the industry in the form of biostimulants. We have been able to corroborate using rhizotrons growing crop plants in the greenhouse that Penicillium melini has a similar effect in the root growth in quinoa in collaboration with Dr. Maria Reguera from the Universidad Autónoma of Madrid. And also we have achieved this positive effect in tomato growing in pots or also in rhizotrons, and most importantly in collaboration with the company of biostimulants Rovensa Next. We already demonstrate in field trials with different species and around the whole Iberian Peninsula that Penicillium melini has a positive effect. For example, here you can see in tomato plants on the weight of the plant and in the yield of tomato. So this encourages us to patent the results that we obtain in collaboration with, in partnership with Rovensa Next as a method for protecting against stress and increasing growth in the plants, the use of Penicillium melini. So with this, we have gone through the discovery of this fungal interaction to the translation to the industry, and but we have a lot to study yet in the field of the basic knowledge and the molecular interaction and how it is affecting the development of the lateral roots. So with this, I finished. Just thank to every people in the lab, mainly the postdoc students and the pre-doc students that are, and the master students that are generating all this data and are working a lot in this research line. Also, of course, to our collaborators, specifically very important Soledad Sacristan, that is another partner of this project. And with this, I finish. Thank you very much to you all for listening. And if you have any questions, suggestions, or even collaboration proposals, are very welcome. Thank you.
Thank you for a very nice presentation of our beautiful story, Javier. So I can already see that there's a question in the chat currently. So thanks for the presentation. How did you find out that Penicillium melini is a good beneficial fungal candidate?
Do you mean how?
Yes, well, yes, through which experiment?
We had a lot of experiments, actually. We can see that in vitro with Arabidopsis plants growing in plates, that we obtain more lateral roots and a higher, a larger main root. And also we have a lot of experiments with crop plants growing in rhizotrons and with the field trials that I showed, where we obtain more yield in the tomato production, and I think Rovensa is also doing some experiments with sugar beets with promising results. So there is a lot of experiments that confirm that Penicillium melini is a beneficial fungi.
Yes, I also saw a raised hand from Salme.
Yeah, hello. I am Salme Timusk from Swedish Agriculture. Thank you, Javier. Very interesting talk and very acute field of study. Actually, also quite interested, like how BGBRs influence foods and which are the actions, what very working mechanisms.
I cannot hear very well. I don't know if you can hear, Naomi.
Do you hear better now?
The connection is not very great.
Do you hear better now? Is it better now?
Perhaps, yes. It is.
Okay, I try to speak louder. So is this like a very kind of questionable thing? Which way do beneficial microbes influence? You actually propose that they influence through main root, lateral root. and seems to be the case. It really works in a particular strains. What we have observed, because we have done major part of experiments in deserts, like in Negev Desert in US, Death Valley, and also in Israel deserts, and Evolution Canyon in Israel, under stress conditions like drought and heat. And we have observed that often what happens is not the main root, neither the lateral root, but root here, which actually plays a major role. So what do you think about that?
Yes, it can be absolutely. We to be honest, we have not centered yet in the root hairs, but it seems to, at least in Arabidopsis, in vitro plates, that we obtain more root hairs, but it's only a visual observation. We have not quantified the growth of the root hairs. But absolutely, in a situation of drought, they can help a lot to capture the water and to help the plants. Yes, in our case, we are more centered in the lateral root development, but for sure it's a thing to see also.
Thank you.
Thank you.
Not sure if there are any other questions. I was also still wondering about something. So you're also still studying what compound potentially it could be for penicillium that is inducing this lateral root formation.
Yes,
do you have any speculation on this?
We have obtained this genome from Illumina sequencing, and we have some putative candidates, but a lot of without any detailed study. And we were waiting to have this genome from Oxford Nanopore that is more accurate. And when we have that we already have, but we have to start to try to understand the putative secondary metabolites that are producing penicillium. So this is what we are going to do, but at the moment we don't have any clue.
Javier, regarding this question, so did you know if this penicillium produce auxins itself?
We know that it is producing, because this Salkowski test, we know that it produces some indolacetic compounds, but we have to do yet by HPLC the analysis to know if they are IAA, the main form of auxins. But what we observe with using the DR5 luciferase line is that we do not observe a huge induction of the auxin signaling. So perhaps maybe it is secreting some auxin-like compound that is activating specifically different parts of the signaling, but specifically in the reverend sites or in the oscillation zone.
Okay. I have another one in your mind. Oh, Poppy also, but just quick, very quick. So you said that it could be an alternative or redundant pathway that, with area and LBD genes. What do you think would be probably, redundancy because there are many genes in the family, or
yeah, there are a lot. We have to try yet with another auxin response factor or perhaps other LBD genes, because as you said, there are a lot participating in it. The R719 and LBD16, LBD18 are the main participators, but there are another. Actually, I don't know. Perhaps there is some, as Naomi suggests, there's some metabolites that is activating directly these downstream effectors. So we have to check it. This is starting, and once that we have the biological system, let's say with penicillium and Arabidopsis and the other plants, we can advance in this field.
Yeah, I have more questions, but we don't have time for that. Maybe later. Okay.
Thank you.
I also still saw a raised hand from Poppy as well.
I put it down because Rafa had exactly the same question as me. If they have a suggestion of what is the bypass of the pathway that activates the auxin. Okay, thank you.
Thank you.
Okay, great. So very interesting prospects there as well. Yeah, thank you once again for a very nice presentation.
Thank you.
And then I suggest we move on to the next speaker.